Welcome to this educational video on recombinant DNA technology. Have you ever wondered how scientists modify DNA to create insulin or genetically engineered crops? Today, together with my co-group mates, we will explore the 6 essential steps that makes genetic recombination possible. Let's dive in. Recombinant DNA technology is a technique in which a fragment of DNA from a donor cell or organism is isolated and then inserted into the DNA of another cell or organism. This allows scientists to introduce a new characteristic into an organism by inserting a new gene into it.
The journey begins with the isolation of genetic material. In this step, the desired DNA is extracted in its purest form, free from other micromolecules like proteins and RNA. Scientists use enzymes or chemical treatments to break open cells and isolate the DNA. This purified DNA becomes the foundation for recombinant DNA technology. Let's say you're trying to create a disease-resistant tomato. The DNA responsible for resistance is extracted from a wild tomato species known for surviving harsh conditions. Did you know strawberries have so much DNA you can extract it with just dish soap and rubbing alcohol at home? Try it! It's like kitchen science magic.
Next, we have restriction enzyme digestion. Restriction enzymes, often called molecular scissors, cut DNA at specific sequences called restriction sites. The purified DNA is incubated with these enzymes under optimal condition to generate precise fragments. This step is crucial for obtaining the DNA segment of interest. For example, imagine creating a glow-in-the-dark fish. Yes, they exist. You can use restriction enzyme to cut the glowing genes from a jellyfish DNA so you can insert it into the fish DNA. Fun fact, these enzymes are so specific, they act like a GPS finding just the right spot to cut.
The third step is amplification using PCR, or polymerase chain reaction. This technique is used to create dozens to millions of copies of the target DNA using the enzyme DNA polymerase. The reaction involves cycles of denaturation, annealing, and extension, exponentially amplifying the DNA. For example, imagine you found a rare gene in a dinosaur fossil. Okay, maybe not Jurassic Park level, but close. So, PCR helps you make enough copies of that ancient DNA to study and understand it better. Here is a fun fact about PCR. So, PCR works so fast, it can make billions of copies of DNA in just a few hours. It's like turning a single puzzle piece into an entire puzzle set in no time.
Now comes ligation of DNA molecules. The DNA fragment of interest and the vector of the plasmid are cut using the same restriction enzyme. This ensures they have compatible ends. DNA ligase, a special enzyme, joins these fragments to form a recombinant DNA molecule. This hybrid molecule combines DNA from 2 different sources, marking the essence of recombinant DNA technology. Example: imagine you're creating glow-in-the-dark plants. You cut the glow gene from a jellyfish and the plant's DNA with the same restriction enzyme. Then, you use DNA ligase to combine them, creating a glowing hybrid plant DNA.
Step 5. Insertion of recombinant DNA into host. The recombinant DNA is then introduced into host cell, such as bacterium, in a process called transformation. Since bacterial cells are not naturally receptive to foreign DNA, treatments like thermal shock, calcium ion exposure, or electroporation are used to make them competent. The host cell incorporates the recombinant DNA and begin its expression. For example, to make yogurt better for your health, scientists can modify bacteria to produce extra probiotics. These engineered bacteria then multiply in the yogurt making process. Fun fact, did you know bacteria don't naturally accept DNA? Scientists have to convince them using techniques like shocking them with electricity. Poor little guys, but it works.
Step 6. Isolation of recombinant cells. The final step is isolation of recombinant cells. After transformation, the bacterial population contains both transformed and non-transformed cells. By using marker genes, such as antibiotic-resistant genes on the plasmid, scientists can filter out the recombinant cells. These cells are then cultured for further use. For example, imagine you're making bacteria produce a protein for medicine. By including a marker gene that turns the successful cells blue, you can easily identify which ones are ready to get to work. Fun fact, think of it as a scavenger hunt where only the cells with a special blue marker win the prize. They get picked for the next big experiment.
And that's how recombinant DNA technology works, from isolating DNA to creating recombinant cells. This groundbreaking technology has revolutionized medicine, agriculture, and research. Thank you for watching, and don't forget to explore more about the wonders of genetic engineering.